And thanks to everybody who's on the call. I'm sure I know some of you, but for those of you who don't know me, I'm Troy Walker. I'm the Water Reuse Practice Lead for Hazen, and I've done quite a bit of work using hazard analysis and critical control points. And I'll talk a little bit about it when I get into the presentation itself. But I really came to grips with the hazard analysis and critical control point process on a couple of projects in Australia, which I'll talk about in a little bit, but they were advanced treatment systems that were built in a hurry in a significant drought situation. And I was working in a state that didn't have a regulatory framework for potable reuse. And so we used HACCP as a way for us to work with regulators to work towards demonstrating, and other stakeholders, and work towards demonstrating that we had treatment systems that would provide protection for public health for drinking. We found it a very useful framework to do that, and ultimately the regulations that were developed were built around them. So fairly simple agenda today. I want to give a little bit of a background to HACCP, where it's sort of come from and what it's about, how we apply it in drinking water and in potable reuse situations. And then I want to spend a bit of time actually working through an exercise of critical control point selection, just so we can see as a group how we might go through the process of identifying where the critical control points are, to get a sense of how that might work. And then lastly, I want to look at how we then integrate that into operations for the long term. In terms of background, in its essence, what HACCP does is it reviews and manages risks to protect public health through a treatment system, and it's very focused on the things that are of a concern to public health. And it's a holistic review that starts at source water and goes ultimately to distribution, the point at which the public would be using that water for drinking. And if I really boil it down, it's really 4 questions that we are asking. We're asking, what are the risks? What are those water quality risks or hazards in the parlance of HACCP that could present a risk to the public? And that boils down to, what are the contaminants of concern that we're going to see, either microbial or chemical, or what are some hazardous events that could result in failures of systems to protect public health? The second question is, what are the right technologies for us to use? In other words, what are the right treatment barriers to mitigate, reduce, eliminate those risks such that the water that we end up with is safe for public health? How do we make sure that those barriers are working? So what monitoring do we use to prove to ourselves and to prove to all of our stakeholders that those barriers are working effectively? And what do we do if something goes wrong? How do we respond if there's a failure of that barrier? And so what does that mean in terms of our operational response? And of course, in plants that we work on in the 21st century, that response is a combination of controls, automation, and operations at that interface. This last comment here is the focus on health-relevant contaminants. It seems obvious, but as we work through the process of selecting critical control points and thinking about critical control point monitoring, HACCP kind of helps us to focus on what is important to public health and differentiate from some of those things that might be important from an operational standpoint. So it doesn't focus so much on making sure you can meet production from your facility, for example. It doesn't necessarily focus on things that could increase the maintenance at your facility, or things that could cause other challenges to you. It's focused on public health, and that's helpful. In my experience, I found that when presenting information on the plant to stakeholders, focusing on HACCP and public health meant that we could take all of the other operational items and issues out of our operational system. They're important, but from when we're communicating to health regulators in particular, here are the things that you need to know to show that we're focused on public health. Now, obviously, there's some interrelationships between those things, but if we focus on the public health parameters specifically, we can kind of distill out of all of our operational information what are the key things that are important to operations and to regulators and to other stakeholders. It's got an interesting history. In 1959, the Pillsbury Company was contracted by NASA to work on helping prevent food poisoning in the Apollo space program. What I failed to mention is that HACCP is something that we've borrowed from the food industry. And you can think about, maybe the risk of food poisoning in a space capsule is fairly low, but the consequence would be awfully high. And so Pillsbury looked at a methodology by which they could ensure that the food products that they provided to the astronauts were contaminant-free, had no microbial risk, and could guarantee that they were safe. It takes a focus away from, if you like, end of pipe or final product testing, and instead looks at developing barriers through a process to make sure that those barriers are working properly to eliminate risks, such that we already know the answer in the final treated water or the final treated product. And the picture that I've got here is actually taken from some of the HACCP material that you would see related to food production. It's well established in the food industry, and if you go to the supermarket in the next day or so, I'm sure you'll glance along the aisles of any products that have been through some kind of processing, very often have an HACCP certification. And that means that that food production facility has looked at eliminating and preventing risks. And those risks are process control related risks, and in the food industry they're often related to things like PPE for some of their staff and procedures around handling of those food products. There's an international standard amongst the ISO set of standards which details food safety management. It's ISO 22000. And a lot of the work that has been translated into water applications has come from this standard and has been adopted. And in many cases, the adoption is as simple as changing the word food safety to water safety, and changing the word, food contaminant to water-based contaminant. When I was working on the project in Australia, we actually went to the effort of certifying ourselves to this standard and being audited against this standard as a means of demonstrating that we were following a critical control point approach effectively. And we'd modified it slightly to be more relevant to drinking water and potable reuse in that particular case. But it was very adaptable. And so a lot of the basis that you see for the subsequent work that's gone into HACCP for water applications is in fact based on things that have been articulated in this standard. It's used for water treatment systems worldwide. It gets a lot of use in Europe, and that's probably not surprising given that ISO standards are commonly applied in a European context. I remember researching this for some of the work that we've subsequently done for some drinking water projects, and of all countries, Iceland actually has 70% of the drinking water, or 70% of the population is served by drinking water systems that are certified to HACCP. It's used extensively in France, it's used in Switzerland, it's used in the Netherlands as well. I'm from Australia, as you've no doubt detected from my accent so far, and it's been used at a number of drinking water facilities in and around some of the major city areas. These first three plants here are large facilities in and around the city of Melbourne. Some of the large seawater desalination plants that were built in Australia in the last 10, 15 years are also certified to HACCP, managing the water quality risks through that process. The Gold Coast desalination plant in Queensland, which is about here, is one of those. In the US, here, HACCP has been considered in a number of studies to determine if it's something that would be applicable to the application of drinking water. It has been used extensively on source control, and I've got an example here from Alameda County in the late 90s, which adopted HACCP principles for managing their catchment and managing risks entering their catchment for source control. Later this month, actually, I'm presenting with the EPA, who have a working group that is in the early stages of thinking of ways that HACCP could be adopted to further enhance safety for drinking water. So that's an interesting space to watch. Now, for what we're interested in, for potable reuse, it is used in multiple projects, both here in the US and worldwide. There's a number of studies that were conducted in the last few years as part of what was then the Water Reuse Research Foundation's Direct to Potable Reuse initiative, and subsequent work, which has initially identified the use of hazard analysis and critical control points. It was then developed into a project that looked very closely at the robustness and the reliability of multiple barriers for direct potable reuse using critical control points. I was involved in this particular project, and what was interesting is that we worked through the application of critical control points, but we also looked in depth at data from a number of potable reuse and other treatment facilities that were using the treatment technologies used in potable reuse, and grabbed their data and reviewed their data statistically to determine how effective were those treatment barriers at removing the constituents of concern, either the microorganisms or the chemicals that we were concerned about for DPR. There is a wealth of statistical analysis that was done in that report, a lot of Monte Carlo analyses around each one of those treatment barriers to really show the level of robustness of those barriers, and then working backwards to illustrate what are the best types of monitors that can be used to validate those barriers. And we'll talk a little bit about that as we go ahead. Those of you that are keeping an eye on California would have seen that they've recently released their draft requirements for direct potable reuse. So they've started to look at the draft regulatory requirements. One of the things that was initially tagged in their first report from the expert panel back in 2016 and has now been translated into the draft guidelines is an explicit requirement of the use of critical control points. They have slightly different terminology. They're calling their control points pathogen control points and chemical control points, just to differentiate between what's important for microorganisms and what's important for chemicals. But they're wanting to use those same principles. Now, I mentioned earlier the project that I got to know and love, critical control points and HACCP, was the Western Corridor Recycled Water Project in Australia, which was a set of 3 advanced treatment systems, membrane-based, so MF, RO, UV advanced oxidation, which were built very quickly. They were built in the space of about, one plant was built in 9 months, if you can believe that, up to about 20 MGD of capacity. The entire system, which was 3 plants and a pipeline connecting them, was built in 18 months with no regulation. And so the operations team that I was leading here on the technical side worked through the critical control point process to essentially form, we were effectively self-performing and self-regulating, if you like, demonstrating using critical control points, getting ourselves certified and audited against ISO 22000 to demonstrate that we were achieving that level of safety and reliability, which then moved into a framework for our regulators to give them time to catch up and help develop the regulations for that particular system. So how do we apply it? What I wanted to do now was kind of walk through some of the specifics and some of the terminology, because I think this is sometimes where we get a little bit hung up and a little bit confused around, like anything, when we jump into terminology, the terminology can be a little bit disabling initially. And so I thought it would be important for us to identify clearly what is a critical control point. And I'm using the definition really that HACCP uses to define it. And I don't like reading words from slides very often, but it's worth going through this definition here so that we have some clear understanding of how this is usually applied. And those control points are the points in the treatment process that are specifically designed to reduce, prevent, or eliminate a human health hazard, and for which controls exist to ensure the proper performance of that project. So it's the point in the process, it's the treatment barrier itself that is reducing, preventing, or eliminating that health hazard, so either microorganism or chemical or both, and for which we have some controls. It's something that we have operational controls, and we have some way to monitor the performance such that we are confident that it's meeting that minimum requirement of eliminating or reducing those risks. And so the example here that I have is reverse osmosis, and I've deliberately picked on a membrane process at the moment. I know that I'm talking to Colorado and that many of the systems that we're going to be looking at won't be membrane processes, but the reason that I'm doing that is because I'm holding the non-membrane treatment processes until later as an exercise that we will walk through to select critical control points for that process. So I'm going to use RO as our initial example, and because it's a fairly simple barrier. So in this particular case, the critical control point is the RO process itself. It's the process and the membranes, because those membranes are the barrier that are reducing chemical constituents, that are reducing microbial constituents. Now, the next thing we have is our critical monitor. It's the thing that is telling us how well this barrier is operating. Is it operating suitably that we have confidence that it's removing our hazards down to the level that we expect? Now, you'll notice that I said the level that we expect. There's a couple of tricks with that, and I'll talk a little bit about that in a couple of slides, but we are always limited in a sense by the degree of sensitivity and the degree of correlation that our particular critical monitor has. In the case of reverse osmosis, we typically monitor something like electrical conductivity, which is a surrogate for dissolved salt removal, remembering ultimately an RO is a salt removal device. Now, we know they also do a pretty decent job of removing microorganisms as well. And in some facilities, perhaps you might see a total organic carbon analyzer there to give you slightly higher resolution. That also helps with chemical removal. What these analyzers don't do is give us a direct correlation necessarily with microorganism removal. And so we have to develop some of those correlations and perhaps not always provide that barrier with the full credit that it might be achieving in terms of removal, because we don't have an analyzer that's sensitive enough or specific enough to give us that piece of information. As we walk through gathering up our microbial log removals for systems, this is often one of the challenges, is how do we know what level of removal we're getting? So in some cases, we're limited often by the monitoring capability across a process to determine what level of removal we're counting on for that particular barrier. Now, if that monitor detects that the barrier is not sufficiently intact to the degree that we can monitor it, then there will be a control action and an operator response that's initiated as a result of that. That might be that we need to take that particular process offline, or we need to divert the water from that process. In many cases, you will have multiple treatment processes in parallel. So if you have an RO system or an MF system or carbon filters, you may have a number of those in parallel. You will be monitoring perhaps each one, which gives you the opportunity to only work on the particular specific filter or RO unit that might be giving you the issue at the time, and take the responses and actions on that. Now, so that we are ahead of ourselves in protecting public health, we typically give ourselves a couple of limits on the monitoring. One of them commonly referred to as an alert. If you think of that in the parlance of alarm conditions, think of it as a warning alarm. We're getting close to our limit. We haven't crossed it yet, or we haven't gotten to it. We might want to take some preventative actions. It might be time for an operator to do some checks, time to take some time offline to do a little bit of troubleshooting to identify what the issue may be before we get to the point that we've gotten to breaching our barrier effectively. Then we have an action limit, which is the next level up from our alert limit. This is where we do need to take some action. Doesn't necessarily mean that we would set that alarm point at an absolute failure condition. Very often we'll set it just below that so that we still have a little bit of room to move. But this is the point at which we need to take a rapid response because we're not confident. We don't think that barrier is going to be operating effectively, or it's very close to not operating effectively. In both cases, this is really operations as usual. We're monitoring our system, we're making sure that we're checking our operations, we're making sure that we're operating within our control limits, and then we're taking action if we are concerned that we're not. And so in the case of an action limit, that may be a shutdown, that may be a divert, and then there will be a procedure for operations to follow to return that safely to service after working through some troubleshooting to identify the issue at hand. So a bit of a long definition of CCP, but I think we got there. HACCP has, I think, 7 principles if you read through it in detail, but in my mind, it really boils down to these: that we assess the risks, we identify the critical control points that we talked about before, which eliminate those risks. We validate and set the limits of operation for each of those barriers, and we have monitors to validate that, to back up that it's operating as it should be. And then we have response procedures, both control automation and operator response procedures, in order to safely return the system to service, take action and safely return it to service if something's gone wrong. Assessing those water quality risks is a matter of identifying for our particular system what are the chemical hazards and what are the microbial hazards. This will be a combination of identifying the levels of contaminants from long-term sampling or historical data or benchmarking other facilities. From a microbial standpoint, that's where we look at our quantitative microbial assessment, determining the levels of microbial risk that we anticipate to see. And oftentimes that level may be determined as a maximum level and a maximum risk, which is where we get our log removal requirements from ultimately. So these are the risks that we look at. But we also need to look at some of the hazardous events that may occur. What are some of the things that might go wrong in our treatment system that we also need to protect against, and having critical control points set up around mitigating some of those. I've got some examples of those to come. Very useful in assessing water quality risks to build risk assessments and build risk assessment tables. And this is a very small snapshot of a risk assessment table that we took actually from one of those reports that I mentioned earlier. This is from the 1303 report. And this is just the first 4 lines of what was a multi-page contaminant risk assessment, looking at the levels of risk of contaminants. Here I'm showing some of the microorganisms. The full list, which if you were to read the report, you could look at, runs through series of chemical constituents as well. And we identify what is the inherent risk that we see in this case in raw sewage, or we may start looking at the risk in secondary effluent. And then what do we need to get to a low risk condition? So what is the level of treatment and the number of barriers that we need in place in order to get to our acceptable level of low risk to provide that water to service? Then we come to determining the critical control points, and I won't labour this flowchart too long, but it's a pretty useful guide that we can use to identify what's a critical control point, and very helpfully what is not a critical control point when we're working through our system. Fundamentally, we take the treatment process and we really ask these three questions. The questions we ask at each process are, is there a hazard at this step? What are they? Can that hazard be controlled at this step? And is this step intended to eliminate or reduce the risk? And if we answer those questions correctly, then if we answer those questions in the affirmative, then yes, this is likely a critical control point. So if we walk through our process, it helps us get there. Is there a hazard here? Yes, there is. Have we got control measures at this point in the process? Yes, we do. Is this step required to meet either log removal, so microbial reduction, or water quality targets from a chemical standpoint? Yes, it is. Then it's a critical control point. But we have off-ramps here. If we go through those questions and find that, well, that process really doesn't meet the requirement for log removal or water quality targets, no. Could contamination occur here to increase? Well, no, then it's not a CCP. So we have some off-ramps. This is a really helpful flowchart for us. If we come across a treatment process, we're not sure if we consider it a critical control point or not. An example that I sometimes use is feed strainers up front of an MF system, for example. Are there hazards there? Yes, there are. Are there controls there? Well, not really. Is this control necessary for safety? Well, we'll have additional barriers downstream of this in order to remove our contaminants. So no, it's not a critical control point. So it's a helpful flowchart to get us to that point of selecting between them. I mentioned earlier, I think when we were talking about monitors, the critical control points and how we validate microbial log removal. And we need to find a way when we're determining the microbial log removal across a particular barrier to how we link that to the critical monitoring. In some cases, we may have some direct correlation. So those of you that are familiar with microfiltration will know that there is a direct membrane integrity test that we can use that can give us log removal for Cryptosporidium and Giardia directly. But there are some treatment processes by which we don't necessarily have that direct correlation. So we use a surrogate measure. I used the example of reverse osmosis earlier, where we don't really have a direct correlation that we're getting the full log removal, but we get at least some minimum based on conductivity analysis or maybe TOC analysis that might give us a log or two. So we know that the system's working effectively, at least to that level. We know from other experience that we can get at least that log removal from that level, but it's not a direct correlation. Sometimes we use it from previously developed data, so we rely on things like CT tables for chlorine or doses from UV system based on established guidelines. And then there are sometimes other accepted principles, like taking some of the rules from the surface water treatment rules for conventional filtration, for example. And I mentioned earlier that in the California draft DPR language, they have parsed out the difference between microbial and chemical, between pathogen control and chemical control. Chemical control and chemical removal, that's usually based on a surrogate measure. Things like electrical conductivity or TOC or UV254 or other indicators that we may use across our treatment process. So that's the critical control point monitor might use some of those. Important to point out that there are other critical control points that might be there to simply prevent contamination as we add chemicals to the process. So an example of that I use is for RO membranes. We typically dose or form chloramine ahead of the RO, and we do that not from a health standpoint. We do that primarily to prevent biological growth on the membranes themselves. But if we don't dose those correctly, if we overdose or if we don't get our order of dosing correct, we can form disinfection byproducts, so we can create a hazard. So while this is used for fouling control on the membranes, which is an operational issue, that's not a health issue directly, the formation of disinfection byproducts does present a risk. And so we identify a critical control point around that process to mitigate and minimize the risk of that contamination. And so if I walk through the RO-based process very quickly, and then we'll get on to the example I wanted to work through of non-RO-based treatment processes, we can identify what are the critical control points. And just ignore this little table over here. Actually, I didn't want to get into a discussion of log removals in this slide, and I meant to remove that, so just ignore that for the moment. But if we walk through microfiltration, it can remove microorganisms. We can validate that with critical monitoring, which is our integrity testing. And so that becomes a critical control point for us. Reverse osmosis can remove microorganisms and chemicals of concern, and we have some monitoring on the RO to at least tell us to some level that we have some removal capability, so it becomes a critical control point. Advanced oxidation, the combination in this case of hydrogen peroxide and UV, we get some microorganism removal across the UV, which we can leverage from our UV dose knowledge and other studies to give us removal. Peroxide UV combined can help with some chemicals of concern. In fact, the UV can assist with some chemicals of concern as well. So, and how do we monitor that? We might monitor that with UVT. With advanced oxidation, there are some specific parameters that we use to ensure that we're getting the right energy doses of UV into that system to give us the validation that we're getting removal. Chlorine is well established with chlorine CT. And so you can see that those are our main critical control points, but there's a couple of additional ones. I mentioned earlier that concern of contaminants coming into the system. And because we dose chloramine for the membrane treatment just to prevent biological growth, that can also form disinfection byproducts. NDMA is one of them. It can form some others as well. So this is a critical control point because we need to reduce and eliminate, the hazard of those chemical formations as much as we can. And we have some monitoring around the way that we dose those systems and dosing levels and dosing feedback in order to ensure that we meet that. The last one here that we considered a critical control point is stabilization of the RO permeate, because we need to ensure that we minimize the risk of things like lead and copper leaching into our distribution system. So in this case, we are again preventing a later contamination into the system. So there's a set of critical control points on that basis. Now, what about the other important aspects of operation? And one of the things that people often ask when we walk through critical control point assessments is, what about scaling an RO? What about fouling a filter? What about all of these things that are important to me as an operator to make sure that I maintain production, that I don't ruin some of my infrastructure or require additional maintenance? They're very important. But we want to separate the things that are important directly for public health for the things that are important for other operations. So the example I show here is with an RO system, we may dose acid and an antiscalant to prevent scaling on the system. That's really important. If we lost those doses for any period of time, we might have a substantial impact to production. We might have a substantial impact to the asset. And so what we have sometimes done in operating facilities is to use a different terminology that we call the critical operating point that goes through the same sort of rigor operationally as a critical control point, but just sits in a different category so that when we're talking to health regulators, when we're reporting to health regulators, when we're reporting to stakeholders on health, we can separate the two. And we don't get lost in the weeds, just so that we don't end up having a conversation or get wrapped around the axle with a regulator over whether we're fouling our RO or not. That is a problem for us operationally, but it's not directly going to lead to a public health concern if we're managing our critical control points effectively. One of the other things that we often talk about is when we get to that interface between the wastewater recovery facility or the wastewater treatment plant and the advanced water treatment facility. In some cases, the point at which you move from wastewater treatment to advanced water treatment is clear. Sometimes it's less clear depending on the treatment processes that you use. But sometimes it becomes a challenge to incorporate the wastewater treatment facility as a critical control point, because what is the monitor that we should be monitoring that gives some correlation of performance for microbial removal and chemical removal? Do we have risks in the raw sewage coming to the wastewater treatment plant? Absolutely. Are we relying on the wastewater treatment plant to achieve a minimum level of performance to operate our advanced treatment? Absolutely. Is it easy to correlate microbial and chemical removal with a monitor downstream of a wastewater treatment plant? It gets more difficult. There are examples where the wastewater treatment or an element of the wastewater treatment plant are used as a critical control point, and there are examples where it is not used as a critical control point. However, in both cases, monitoring at the end of the wastewater treatment facility is still valuable and is still used in order to provide some certainty and surety to the operation of the advanced treatment facility. So we may, for example, monitor ammonia to demonstrate that nitrification is working effectively in the advanced treatment system. But can we guarantee a log removal of microorganisms as a result of that, or a chemical removal? It's a little more difficult. So one of the things that I have seen in a number of occasions is the use of another term called something like a quality control point, where we want to make sure that the quality leaving the wastewater treatment facility meets at least these particular parameters. It might be turbidity from a clarifier. It might be ammonia if we have a system that's nitrifying. It might even be nitrate if you have a denitrification facility there. Monitoring of those downstream works very similarly to the way that you would operate a critical control point monitor. You may have alarms, you will still have control actions, but you may just not rely on it as a critical control point for your for counting your microbial removal or for counting your chemical removal. It's a bit of a subtlety. So the last sort of 10 minutes here, or even faster if I can go through this quickly, I wanted to walk through an example of critical control point selection for a non-membrane treatment facility. So a carbon-based treatment facility. And this is based on work that we also went through on the 1303 report. And I've just got a simple example here of a flox, SED, ozone, BAC or BAF, GAC, UV reactor, and chlorine. In this case, it's set up for direct potable reuse. And so walking through the exercise of determining what the critical control points might be for this facility. So I'll work through it very quickly. What I've done here in the tables, and you'll get a copy of this at the end of the presentation, and you'll get the recording as well. But I've walked through and parsed out these questions for each point in the process. So as we walk through, what's the first step in our process is coagulation settling. Well, is there a hazard at this step? Yes, there is. There's microorganisms and chemicals, obviously, at this point. Do we have good controls, though, for those microorganisms and chemicals? Well, we can get some settling, and we'll probably get some microbial removal settling out with our flock, but not good enough that we could really correlate a removal just at this step. So is control necessary here? We've got processes later. This is not considered a critical control point. So we move on to the next step in the process. Ozone. Is there a hazard? Yes, there is. Do control measures exist? Well, yes, we can set up the ozone system to operate with a CT, with a confirmed concentration and a residence time. So yes, that can be a critical control point if we choose to operate it with ozone, residual monitoring, and flow monitoring. So what's our monitor? These things. So yes, that's our CCP. I move on. Is BAC a critical control point? We've got the risks of microorganisms and chemicals still. We haven't removed all of them. But do control measures exist? Well, without ozone being considered as part of the process, or for that matter, if we don't include the flox SED as part of that process, well, on its own, no, because we don't have either the knowledge in this process that we have coagulated, flocculated solids from a filtration standpoint, and we don't know on its own that we have ozone to oxidize the organics into a state that they would be well removed on the BAC. Now, if we combine the ozone and the BAC and consider this as a process combined, consider this as a CCP together, then yes, because we have controls and we have monitoring for this process combined, in this case, to remove the risk of some contaminants, some chemicals of concern. In the same vein, if I then combine the floc-sed with the BAF, the ozone just happens to be in the way, then yes, similar to the way we would accept it for the surface water treatment rule, that combination of floc-sed and filtration can give us some microorganism removal. So combining processes together as a combined barrier is an approach that we can take in developing our critical control points. And what are our monitors? Well, turbidity, perhaps our coagulant dose going into that treatment process. And then lastly, GAC. Yes, we can get some removal of chemicals here. We can monitor carbon life, perhaps TOC or UV transmittance across the system. UV is well established for UV dose, and so on in developing our critical control points. So that was quite fast. And when you walk through these critical control point assessments, it's often done in a workshop setting, and there's a lot of discussion around how you include a barrier, whether you don't include a barrier, what's the best monitor to use, and then the next steps are, what are the limits that we would use around this treatment process? How much log removal do we think that we can achieve? What do we use to monitor that and verify it? And then ultimately, what are the limits that we need to operate to in order to achieve that? So lastly, I just wanted to touch on the importance of the operations interface from this critical control point process, because ultimately it's the operations team that takes custody of this system and operates it through the life of the plant and is responsible for the maintenance, for the operations, for the monitoring, for the regulatory reporting of these systems as we move ahead. And developing well thought through control and operational response procedures is really important. This is an oversimplified response to, I've used the terminology critical limit here, where something moves from critical operation and we get into a critical condition because our action limit might trigger. And it's what we do for each one of our critical control points, each one of those treatment barriers, is develop a response procedure which considers controls and which considers operations. And it looks at a combination of who needs to be notified, what are the procedures that we need to follow, what's the critical, was it real? Do we go and verify our instruments and monitors to make sure that we validate that it was a real condition? We work then through what do we need to do to return that system safely to service? What sort of investigations do we need to follow? Who needs to be informed? Which stakeholders from a regulatory standpoint, from an operations standpoint, and so on. This is a simplified version, but at the plant that I was working on in Australia, we had these response procedures in flowchart form laminated at each one of the control desks with very specific requirements, and then if there was a reference to another specific operational procedure that needed to be followed, they would be followed. So, you know, think about the astronauts as the red light comes on in their capsule and they grab for their folder and they grab for what is the operational procedure that needs to be followed, and that's effectively what we had in place. The other thing that I didn't mention here and should have is that one of the really useful things that I found operationally was on our SCADA screen or our HMI screen was that the default screen at each of these plants was just a list of squares with each one of the critical control points and a condition. Green, yellow, red. You know, green is operating correctly. Yellow might be in an alert condition. Red would obviously be in an action condition. Something that just really demonstrated not only to the operators on the site, but to any stakeholder who's coming in to watch, that critical control points and the protection of public health is front of mind and is paramount to the operations of that facility. Lastly, of course, we don't want to, as good operators, be relying on waiting for the alert condition to occur, waiting for the action condition to occur. It'll happen sometimes because some failures are more immediate. Membranes can fail, filters, laterals can break, analyzers can fail. But monitoring and good dashboard reporting are really good ways to, and it's important to make sure that we have that data front and center and in a timely fashion so that operations know where they're operating, know where they're running, and can anticipate as much as possible when any of our critical control point barriers are moving towards less amenable conditions. The last thing that I will say, and I should have put a slide on this and I didn't, and that is that one of the things as you think about this, you're reliant in operations as much on the analyzers and the ability of those analyzers to detect a problem with the barrier as you are with the barriers themselves. If you think about it, a true failure of a critical control point system is not if the barrier has some kind of breakthrough, because we've got monitoring to detect that. We've got controls and operational responses to respond to it. The true failure is if our analyzers fail and we don't detect that. And so a lot of focus on calibration, validation, and maintenance of analyzers is very important. And I think that is the end of the presentation. I'm sorry I haven't left a lot of time for questions, but I'm happy to stay on a little longer if anybody would like to answer any. So back to you, Tyson.
Yeah, I think we just open it right up. It was excellent information. Thanks. I'd like to hear people's thoughts or questions, concerns. But just reiterating, we would like to use this model. We think that some of this is intrinsically built into the pathogen monitoring surface water rules, which would then translate to direct potable use rules in the pathogen sense. But the chemical monitoring is sort of brand new to drinking water regulations in Colorado. And we thought this was a compelling, in the operations work group, approach to make sure they're in compliance. Each system using this and reporting monthly to the state. Again, not if they hit alert limits and action limits, that's not a problem. It's if the actions weren't taken to deal with it, that's the problem.
Kevin, you've got a question.
Yeah, I had a question, Troy. Thanks for the presentation. Great stuff. My comment is, I guess I'm having a hard time understanding why a biological filter would not be a critical control point.
So you're talking about a BAC on its own?
Yeah.
Yeah, in some ways it's semantics, I guess, and it's because we're talking about, do we have controls? And if you take the biological filter on its own, it's in the context that I showed it on that treatment process, it's assuming that you've had an ozone dose ahead of it to pre-treat, if you like, or to oxidize the organics in that stream such that the BAC operates effectively. So in the context of does BAC on its own work, we thought it was more appropriate to consider the process as ozone BAC together.
Okay.
So that becomes, in the same way that advanced oxidation might be peroxide UV or something like that. It's a way of saying that those two, because in order that process is working effectively, at least in that context, it is an ozone dosed feed that is coming to the BAC. And we might want to monitor the ozone dose as one of our critical monitors to ensure that it's operating correctly. One of the things I will say is that when you walk through the workshops to develop critical control points, these are exactly the discussions that you have. And if I went to a number of different, and I can think of ones I've even worked on, a number of different treatment plants and processes where we've gone through this exercise, you sometimes have slightly different conclusions. And it's not, it's not cast in the 10 Commandments quite. I think as long as you're aiming to the same goals. That's just the approach that we took in that particular situation. I mean, there are plants, for example, that consider the wastewater plant a critical control point and get log removal for it. There are others that don't.
Okay.
So there's a bit of room to move. Yeah.
Thank you, Troy.
No worries.
Troy, this is Doug. Question on redundancy, I guess. When you you kind of prefaced the presentation about not looking at log removal credits and things along those lines. What I'm wondering though is you've got critical control points and the idea that do all of them have to be operating and in the green, if you will, if you're looking at your dashboard, to say that the facility is meeting its goals, or is it a way, again, the critical control points is a way of assessing that. So I don't know if that question is clear, but I'm kind of worried from an operational perspective, that monitoring something and it goes to alert, and am I shutting down my entire process? Can I rely on other barriers along the way, and how these things interact?
Sure. I'll tackle that a couple of ways. So the first question is, if you have an alert or an action, do I have to shut down my whole process? It kind of, in some ways, that depends on how your plant is configured. And so I give the example of, let's say you've got membrane filtration or just filtration in your system, and the turbidity on one of your filters has gone high. You can action that specific filter and not necessarily have to shut your entire plant down. You might shut that filter down, do what you need to do to return that one to service or divert the water from that particular filter. You don't need to shut the entire process down. If you had a part of your treatment train where, and so that just becomes like redundancy. It's like, do you have a duty standby pump? Do I have a duty standby filter? If you have that duty standby filter, you can manage it the same way as if you had a failure in a pump, I would say. Now, in terms of redundancy for additional processes, so in other words, if I have to meet 12.10.10, am I just meeting 12.10.10 and nothing else? That's a good question. I think we saw recently in the California draft DPR regulations, they have taken an approach where they have quite explicitly thrown in some additional log removal they would like to see as redundancy. The thing you've always got to remember with a log, of course, is that's 90% additional removal. So I think they asked for 4 additional log removal. It's 99.99% removal again on top of what they had. So I think it comes down to a question of how is the log removal risk assessment identified in the first place, and how much, so how did we get to our 12.10.10 in the first place, and what's that level of risk? Do we want to invest in some additional barrier that's available to us in order to get to our 12.10.10? So you can always make the decision as a designer and an operator to have some additional redundancy. The other thing that we found when we worked on that 1303 report, which was really instructive, is that we very often are grossly under crediting ourselves with the removal capability of some of these processes. And that's because we don't have the sensitivity or the correlation available from the critical monitor that we're using. RO is a good example. Challenge tests would show that we confidently get 4 to 6 log removal of microorganisms across an RO system in most normal operation. In fact, I've seen studies where a fully oxidized RO membrane is still getting 6 log removal of microorganisms. But we don't have a monitor that can show us that. We have a monitor that can tell us, well, you're operating the RO well enough that you can probably correlate 1.5 to 2 log removal of microorganisms across it. So there's quite a bit of redundancy in that space, which is in the undercounting. Does that help answer the question?
Yes, thanks.
Right. Well, if anybody has a question later on that they are thinking about, you can see you're more than welcome to send me an email or give me a call on the cell phone. I guess I haven't used the main office for a while, and I'd be more than happy to get back to you.
I really appreciate it. I think it was instructive, and I think, yeah, it's important for all of us to keep in mind. Again, the state has pretty decent, well, at least we're considering it, pretty decent framework around pathogens because we're basing our entire surface water treatment rule on the way to calculate CT tables, and which is implicit. But the chemical, again, is chemical removal barriers are, I think is going to be a conversation with each DPR installation, and I think this is the model we need to use. So what I would like people to keep in mind is as you look at the regulation throughout September and October, and as we compile it, this presentation will be a key reference of like, this is what we're talking about, but also then, do you think it's too open-ended or do you think it's not open-ended enough? These are the types of conversations, because we don't have 6 DPR plants in Colorado that we can say, these are the ones we want to talk about. We have them other places. We have California, we have Australia, but we really do want to allow this conversation because we think we will get a bit of variability to different utilities using different carbon-based treatments and different types of advanced oxidation. So we want to be able to have these conversations more open-ended than to say, thou shalt always do this dose of hydrogen peroxide or something, which is usually how regulations are structured, right? And especially in the U.S. and Safe Drinking Water Act.
Yeah.
And this is a mechanism where I believe we'll be able to be a little more flexible.
One of the things that I found personally is in an operations context that was so useful with critical control points was ensuring that the focus was clear when talking to regulators. And it took, any other operational items or issues that we may have had that were not health related directly were not something we would necessarily get confused about or we'd be having long discussions with. We wouldn't be talking to regulators about whether our membranes had failed or not. Yeah, maybe over a beer, but not as something that was going to get in the way of our permit. And I think the ability to parse out and demonstrate that clear focus on what the health parameters are is pretty useful.
Well, thanks all. I appreciate everybody attending today. Again, Troy, I'll work with you about this recording and where you guys were okay with us posting it, et cetera, for people's reference.
Absolutely. No worries. Thanks, Tyson. Thank you, everybody. And if I haven't met you already, hope to do so before too long.